here you see where the study started many years ago. We were trying to understand how Semliki Forest Virus and small enveloped RNA virus enter cells in tissue culture. The surface of the cell is shown here by electron microscopy, and first the features include the filopodia. These are long actin containing extensions of the plasma membrane, and what is here is probably a lamellopodium, another common structure present on cell surfaces. But most importantly, these small spots that you see are viral particles attached to the cell surface, and some of them are being internalized in invaginations, such as here, and there are other ones. Up here is one where a viral particle is disappearing into the surface of the cell in a deep invagination. What happens is that the virus is being internalized by endocytosis. The bound particle first is taken up into clathrin-coated pits, and these invaginate, forming clathrin-coated vesicles, and using this standard pathway of endocytosis, the virus is delivered to an organelle called the endosome. And in this endosome, the virus is exposed to a reduced pH around 6, and that induces a conformational change in the spike glycoproteins, resulting in the activation of membrane fusion between the envelope of the virus and the limiting membrane of the endosome. As a result, this icosahedral capsid with its RNA genome is released now into the cytosolic compartment and almost immediately uncoated. That means that the capsid falls apart, the viral RNA is released, and then it is used here as the messenger RNA for the synthesis of the first viral proteins. This is how it looks in an electron microscope. You see the first step, virus being internalized in a clathrin-coated pit, which has this electron-dense material on the cytosolic side. These are clathrin-coated vesicles. And in some cases, one can actually see the capsid down here being released from an endosomal structure before it has had time to uncoat.